Hello, William. Ah, so what's new? Let's see. Hello, Nathan. Uh, okay, so let me ask somebody else what their plans are. Um, okay, so Catcher, what are you doing and what do you plan to be doing in the future? You there, Catcher? Ah, hi.
Oh, yeah. So I was connecting to my AirPods. So I'm actually undergrad right now, but I do research in synthetic biology. And I'm gonna pursue a PhD in systems biology, looking at like developmental processes in the embryo. And hopefully
Cool.
we'll go into academia after that.
Yeah, so Catcher, you're in a lab right now. That
I
seems
am.
obvious. What are they doing in the lab right now?
Yeah, so we use synthetic biology to kind of try and harness developmental processes to engineer tissues. Right now it's all very early, but we're trying to do stuff that will be like proof of concept for down the line, like think of like the Spider-Man movie, they're making like lizard tails and like regenerating arms, like obviously for a better purpose than making lizards, but yeah.
Okay, so what defines synthetic biology and systems biology for me and presumably the others?
Yeah. So synthetic biology aims to well, there's a couple different branches. The larger synthetic biology community usually uses bacteria and they like genetically engineer bacteria. So they play with the DNA of these bacteria to reprogram them and do what we want. And that, yeah.
So I know there's been an effort to sort of make a minimal bacteria
Oh,
where you sort of
yes.
pull away as much as you can and still have it wiggle around.
Hmm.
Um, and that is done in the, I think, as an effort where now you could add stuff to it and have a better sense of what might happen. How's that going?
Exactly. Yeah. Yeah. So there are a couple labs at USC that are actually pursuing that as an end goal. Um, I think I don't know, that's not my like very specific expertise, so I'm not exactly sure like what the timeline on that is, but it seems like good progress is being made. That would be once we get that, I think that will accelerate a lot of progress because, like for example, one goal we do is we work in mammalian cells, and we try and usually biology, you look at what's necessary, like you take away components, and that's very like different from the engineering perspective where you're kind of looking at the components and how they work together and then how they generate this system. So with synthetic biology, like what we do in mammalian cells is we're trying to build these tissues so we can understand from like first principles what is making them make, like for example, like make an elongated structure in some organisms and so on. And a
So,
single living
oh,
cell would be perfect for that.
gotcha. So there's synthetic biology and systems biology.
Mhm.
Um, the synthetic is you're actually trying to get bacteria to do something,
Yes.
and the synthetic is or the systems is what?
Yeah, so the systems is that's what I'm interested in doing in the future. And it's pretty interrelated with synthetic biology, but it kind of takes a different approach to traditional biology in that traditional biology in the 1980s and even before that, we kind of looked at these singular pathways and these singular processes in cells and kind of we took out this one gene and saw the effects of it. And lately in the past decades, we've realized that it's not as simple as that and that these cells are just so complex and there's all these different systems that are all connected to each other. And so systems biology kind
Yes.
of tries and it integrates like transcriptomics data, integrates all these different like levels of experimental data to try and get
So,
a sense of the bigger picture.
so it sounds to me like it's largely a theoretical endeavor. You learn a lot about as much as you can about
Yeah.
biology, and then you try to sort of draw arrows on what's connected to what, and later you might intervene in those arrows to
Yes,
create other stuff. So it is
exactly.
mostly a pencil and paper discipline, the synthetic biology. No, the systems biology. Is that
Yeah,
right?
I would say uh I was agreeing with you up until the pen and pencil thing because we use like computers, to do these make these models like you were talking about like the theory.
Yes.
So that's a big so yeah, like pen and paper in a theoretical sense, but um yeah, like another lab I work with, we try and model like these gene pathways and obviously the computers can do it way better than we can with just like our simple little brains.
Yeah.
And then to actually validate that, like you were saying, we then go in and be like, okay, so our model, we predict this, and then we can test it in the cells. And if it, follows, then our model is useful and we can hopefully learn some other things that we can't typically learn by just playing around with the cells in the lab.
Yeah. It sounds very interesting. Really, it
Thank
does.
you.
And um, later, well, today we're going to say more about DTD humans, and then we're going to go back to it's alive, okay? Because we didn't get very far into it. And then I think I'm going to turn to Catcher to have a discussion about RNA. Okay.
Yeah, sounds
Uh,
good.
yeah. Anybody else got any remarks or comments or whatever it is you have? All right. Um, well, then let's turn to the book, and I'll start to share. So we were doing. We just figured out. I asserted that we're robots, right? And so the picture, two robots. Okay, but now we want to look at your set of preens.
Your preens. In this chapter, I will describe some of your preens and how you acquired them. Your most important set of preens comes from your parents. These are the genes stored in the DNA of the zygote from which you grew. I'll call this your innate gene set. During the prenatal period, your innate gene set will build you. Among other things, it will make your brain and provide it with an initial set of programs that are roughly analogous to your computer's operating system. It will also create your sensors, actuators, and emotions. Once you are born, your innate gene set does something surprising. It invites other preens to share control of you. As we know, preens are not noted for their generosity, so why does your initial gene set do it? We will address this in the next chapters. Discuss.
Okay, so you have this innate gene set that is responsible for building you, providing you with a brain, a lot of stuff. But um, once it provided you with a brain, that brain, obviously in humans, starts to control some of our behavior. And since genes in the innate gene set are selfish, they don't even like each other, right? They're competing with each other. Why would they surrender some control of the organism they all intend to exploit to this new stuff in the brains, which in our case is called memes. Uh, why would they do that? Oh yeah, Ryan.
I would assume it's to increase expected survivability of the organism, which would then increase expected propagation of these preens.
Yes, and so that's my view as well. It's got to be a trade-off that they're getting more from it than they're giving away, right? There was this discussion, I guess we already had about um, this being like a synergistic relationship, and so we did discuss that. Um, how is it that the brain could help an organism survive? I mean, in our genes, for example. I mean, what mechanisms are actually involved in that? Oh, William, yes, please. You had a comment.
To add on to that, the reason why the brain needs to acquire memes is because it can't do everything in its lifetime, right? It needs to acquire new memes while it's surviving to better adapt
Okay.
to its environment, right?
Well, wait. So I want to explore this a bit. Okay, so you're saying, wait, so the human genome decided to build this computer and then surrender some control of us to that computer, which because the genes are not generous, I'm curious why they would do this sharing and of control. And uh, it's clear that I think that the genes must have benefited because they did surrender this control to the what turns out to be the memes. Okay, so now what is your remark, William?
Is that in nature there's many niches. Nothing can do everything essentially. So
Yes,
because
that.
the genes can't really maybe adapt in its lifetime of the organism needs to adapt to memes while it's learning.
Oh, okay. So now I think I get it. You're saying that there's an advantage to memes in that they can be passed much more rapidly than genes can. Genes largely get passed when reproduction occurs of the organism. But that, is taking every 20 years. And memes are passed much more quickly than that. Is that part of your view, William?
Yeah, it's a good idea.
Okay. And I think that is an important aspect of the difference between the two, between genes and memes. It seems certainly true that memes can be passed much more quickly and also more broadly. I mean, you can tell somebody a good idea, you can tell 30 different people in a day. Uh, you can't go reproduce a new human being 30 times a day. So I think that's a good observation. Um, I guess the remaining question is what is the remaining question? Um, so it does raise the question though, what is the value to the genes of having these memes running around? I mean, uh, here's a form of the question. Is it just because brains and memes react more quickly, let alone reproduce more quickly than the genes? But they react, they can cause the uh, person to presumably move out of a room if it's on fire, where the genes really don't so directly have that, or to call a doctor if someone's injured. Okay. I sort of have a sense that the main thing that memes do is social interaction. But okay. Let's leave that question and go to the next chapter.
Welcome to the Bacteria Hotel. When you were born, there are about 5 trillion cells in your body, each of which stores your innate gene set. There are lots of red blood cells as well, but they do not contain DNA. Almost immediately after birth, you will become infested with microbes. For example, you will have about 40 trillion bacteria in your gut, each with its own gene set. Current estimates suggest that these bacteria are representatives of about 10,000 distinct species, NPR, 2012. So, your innate gene set ends up sharing your body with a very large bacteria gene set. Your innate gene set has designed your gut to be a full-service bacteria hotel where tenants have a nice place to live and plenty to eat. Why would your innate gene set do such a thing? The answer is, of course, that most of the bacteria are paying guests. They pay for their room and board by providing a variety of services that help your innate gene set. For example, some bacteria contribute by helping you digest some of the food you consume. Not all bacteria are helpful. There are harmful bacteria that, should they get a room, will consume resources without providing services, cause sickness, or in other ways be deleterious to your innate gene.
Okay, but this gives us a hint at why the genes have decided to share control with the bacteria, because the bacteria benefit from the same organism that the genes are trying to benefit from. And the answer is that the bacteria are paying guests. They provide services back. So whoever brought up, it might have been Ryan, this idea of a symbiotic relationship. I mean, here's an example of it, right? The bacteria contribute to digest some food you consume. They also do other things. And in return, the humans let them reside in the organism and use some of its resources. Okay.
Not all bacteria are helpful. There are harmful bacteria that, should they get a room, will consume resources without providing services, cause sickness, or in other ways be deleterious to your innate gene set. Since your innate gene set builds the bacteria hotel and the systems that operate it, it gets to put security measures in place to aid in getting helpful bacteria in and keeping harmful bacteria out. For example, the genes keep the hotel hot, 98.6 degrees Fahrenheit, 37 degrees centigrade, and highly acidic. As a result, though all manner of bacteria enter the hotel when you consume food, most do not succeed in getting a room because they quickly die in such conditions.
Right. So if there's some bacteria that makes its home in fish, and there almost certainly is, then they have evolved to live in fish, which have a body temperature way below that of humans, presumably somewhere around above the freezing point of water, but not too far above it. And so the bacteria that live inside those fish, you throw them into, they've evolved to optimize being in that particular temperature environment. You throw them into something like 98.6 degrees, they're unlikely to survive. It's not impossible. I think there are some that can do that, but by and large, they're not a threat. Okay. It's probably the case that, chicken is a source of bacterial poisoning for humans, but it's probably not chicken bacteria that we get. It's bacteria that are living in the environment and the chicken's been exposed to it.
Your innate gene set appears to use a trick to help assure that the initial hotel guests will be helpful. It has made it easy for maternal bacteria to pass into the baby's hotel during birth. Since the mother has already survived and reproduced, there is a good chance that most of the maternal bacteria are helpful. Once the rooms are initially filled by helpful bacteria, many harmful ones are kept away because they cannot compete with the current residents for a room. This is one way that the bacteria in your gut act as part of your immune system.
Right. So this is an important trick, and we're going to see it again. When the baby comes out, it's sterile, doesn't have any bacteria. But of course, it's going to enter a world full of bacteria, so it's going to get some. So where's it going to get these bacteria? Well, it could just get them any place, but instead, what happens is during the birth process, the baby apparently picks up bacteria from the mother. I mean, the very birth process pretty much ensures that. And since the mother has survived and reproduced, that's pretty good evidence that her bacteria are helpful to humans and not harmful to humans. And so as a result of that, the rooms are filled with what are likely to be helpful bacteria, and many harmful bacteria are there, are kept out subsequently because they can't compete with the current bacteria in the gut. All right.
As an aside, ensuring that only helpful bacteria reside in the gut has recently become a concern of medicine. In the emerging field of probiotics, in a manner resembling maternal-to-baby transmission, helpful bacteria are imbibed in the hopes of improving gut biota to combat disease. Bacteria have access to all of our external, in the mathematical sense, surfaces, including ear canals, lungs, and sinuses. As a result, in addition to the bacteria hotel in the gut, there are hotels in the ears, lungs, sinuses, etc. All bacteria hotels operate on the same principles, and guests in each must pay for their keep. Since the environment of the ears, sinuses, lungs, and gut are very different, it is likely that the bacteria that reside in each are also different. It would not be surprising to find that each hotel has bacterial guests that contribute to our well-being in a variety of ways. So, if the ideas of probiotics prove worthwhile, perhaps they should be extended beyond the gut. Might people with chronic ear infections, chronic sinusitis, or chronic bronchitis be treated with sprays containing helpful bacteria and other biota?
Okay, so what is your opinion on probiotics? Is it been established to be a good thing, or is it just a fad? Does anybody know what the current state is?
There's been a lot of research on it,
Okay.
and it seems like it's pretty well established. There's some studies that have been retracted because like you're using massive data sets, and so sometimes basically they just make mistakes with the computational methods. But there have been so many like studies recently that it seems, and for like years and years and years, that it seems like it's pretty well established.
Okay, thanks,
Yeah.
Catcher. Another question though is, have people extended this to consider the biota that exist on, our bronchi, in our ears, all over us, all our external surfaces, and considered probiotic approaches to those? I mean, are people, huh?
I was just saying, honestly, I'm not sure. I know the stomach, you know, and
Yeah.
the gut microbiome is hugely researched, and skin too has been pretty well researched.
Mm.
But other than that, I'm not too sure, honestly.
Oh, okay. All right, thanks. Um,
Your innate gene set built your bacteria hotel, but it is surrounded by other properties with do not enter signs. For example, your innate gene set does not want bacteria in your blood, so it has built an immune system that among other things will punish trespassers from the gut. Do these security measures work? Absolutely. Do they work absolutely? Absolutely not. Most of us will from time to time get harmful bacteria and experience food poisoning. Some of us will get bacteria in our blood, septicemia, and die. Lecture 19, 20250331 Part 3. Welcome to the Memes Hotel.
Okay, so the point of the last chapter was to examine the gut because it's an example of sharing control of the body with something not in you, with preens that are not in your genetic preen set. Okay, so and now I think there's parallels to what's going on in the brain.
Your innate gene set also built a hotel for memes, the brain. The memes hotel has much in common with the bacteria hotel. When you were born, your brain had lots of unoccupied rooms, that is, locations and memory devices, but soon after your birth they began to fill with memes from the surrounding environment. As with the bacteria hotel, there is a big security problem. There are harmful preens out there that, should they find a room, could be deleterious to your innate gene.
So what's an example of harmful memes that could be deleterious to your innate gene set? William?
Reproduce gene meme, I mean.
No, that's really a genetic thing, isn't it? I mean, at its most basic level. I don't, that's not what I was going for. Okay. All right, Ryan, you got an idea on this?
Something that too far contradicts the preens of the culture that you live in.
Oh, well, that's an interesting way to go about it. Well, let's get back to that, Ryan, because I had a different idea, which was if you get certain memes in your head, they might result in suicide, substance abuse, imprisonment, ostracism. Yeah. I mean, people commit suicide at least partially because of what they've acquired, what their thinking is. So that's an example of what I would call acquisition of harmful memes. But you're saying, okay, you said it's getting memes that are in contradiction to, I guess, societal memes you've also acquired. Is that it?
I guess part of the problem is you may or may not have already acquired them, but if you sort of pick up memes that are that contradict what the cultural preset of your like local culture dictates, then that can be problematic. So think about like Galileo, or anyone that tries to be like secular in a very religious society.
Yes, yes, that could certainly be detrimental to the genes. Um, yes, so you pick up memes that put you in conflict, internal conflict with societal memes or other memes you've had, which are dictating other behavior than the ones you now, these deleterious ones. I think you're right, Brian. I've been thinking more about emotions recently and what they're all about. And what you said that you pick up deleterious memes that are contrary to cultural memes you might have picked up, I think is right. But maybe it's the case that emotions mediate these things. I'm not sure, but it's a good remark. Okay.
Since your innate gene set builds the memes hotel and the systems that operate it, it gets to put security measures in place to aid in getting helpful memes in and keeping harmful memes out. Some of these measures were also used to secure the bacteria hotel. For example, your innate gene set has designed your brain to readily acquire memes from your parents when you are young. The fact that your parents survived to produce you is good evidence that the memes they pass to you are likely to be helpful. Among other things, these memes may help you to avoid future dangers and ensure that you will fit into your social milieu.
So I think that's sort of consistent with your point, Ryan, that so your parents just are going to start to transfer memes to you. And a lot of those will be societal, cultural memes. And uh, that's just like when the baby's born, acquiring all these bacteria from the mother. The reason that that's favorable is because the mother's alive and is reproduced. And again, the fact that the mother's alive and is reproduced is also suggests that her societal uh, uh, memes um, work. And so they're also transferred to the child, the newborn baby or the growing child. Um, and they do ensure that the person typically gets a lot of societal memes from the mother, and that assures that they're not going to have the conflicts or lessens the chance um, that they're going to have the conflicts that you were suggesting, Ryan. Okay, so
For example, if you are brought up a Christian, it is unlikely you will later convert to Islam, and vice versa.
so once those hotels are occupied, uh, say by Christianity memes, it's very unlikely you're going to convert to Islam. And there's emotional reasons why that's the case. Uh, I guess we'll get to that later.
Versa. Parental memes, even those that are not passed directly to you, will influence the memes you acquire while growing up. Your parents will choose your schools, places of worship, what you can watch on TV, and more generally which societies have access to your brain. As an aside, since your parents will choose what and where you eat, their memes also help to secure your bacterial hotel. Eventually, many memes will become residents of your meme hotel. For example, many cultural preens from religions, governments, political parties, companies, and other societies will succeed in obtaining a room. How your brain captures.
Okay, and we've done this chapter before. All right, so any comments on all that? Um, Something? Okay, so now I want to turn to our leftover discussion of it's alive. What does it mean to be alive? Okay. Okay. Um, so this began by saying that there were presumably these self-reproducing molecules, and maybe they were the beginning of life, whatever life means, on Earth. And I was asserting that whether or not you think that those replicating molecules are alive or not, they apparently appear to be involved in a struggle of survival. And that evolutionary struggle has continued through time and has become the struggle that all living things are now engaged in, including you and I. Right. So that's the thesis I'm putting back. But then the question arises, did that evolutionary structure begin with self-replicating molecules, or was there something that came before that? The struggle may not have begun then. And okay, so now we get to what hopefully is interesting.
I will argue that it began far earlier.
All right, so at this point, we're going to bring in Ketcher because we're going to need to talk about RNA like early Earth. So Ketcher, can you sort of join me here? And
Yeah, I'm here.
okay, so thanks. Now let's talk about RNA. It looks like pretty much DNA. It's chemically slightly different. But by and large, it looks like a string of pearls that are of one of four different possible colors, and they're strung for however long they're strung. And that's a piece of RNA in its so-called primary sequence, where you're just told which ribonucleic monomer is in the chain, what colors are being used. Okay, but Ketcher, what happens once the RNA is actually inside a cell and you've got that primary chain? What should we expect to happen next?
Yeah, so you're talking specifically about messenger RNA. So there's a ton of different types of RNA, but if you put this messenger RNA into a cell, then it will bind something called a ribosome, which is a protein. And then that ribosome will decode the RNA and produce the protein that corresponds to what the genetic code is.
You're correct, but actually that wasn't what I was talking about, messenger RNA. I was
Okay.
more talking about an RNA molecule in general.
Mhm.
What will it do if you throw it into, let's call it salt water?
Well, sorry, I think I can answer this.
Yeah, Martino.
It'll fold according to thermodynamically favorable processes. So in the case of RNA in an aqueous environment, it'll form a helix, and it'll form a single-stranded helix.
Well, yes, it actually will, but a single-stranded helix is a little tough to grasp because, when we have single-stranded DNA, it usually finds another single-stranded DNA to form a sequence with.
Well, this has
But
to do with the
I,
stability of RNA relative to DNA, which is enabled by the fact that DNA is more stable due to having one less hydroxyl group on
yeah,
the sugar.
yeah.
But yeah, RNA is less stable than DNA, which is why like organisms like ourselves, we've evolved to have our genetic information stored as DNA because it doesn't decompose as quickly and it's more stable in aqueous environments. But yeah.
so I think
Well,
RNA would actually get degraded first because it's very like unstable, and if you put it into a cell, for example, it would just get degraded by RNases. I guess that goes with some of what Martino's saying.
yeah, so nothing is wrong, but I was looking for a simpler answer. That necklace, if you put it into a cell-like solution, I'll call it salt water, will in fact uh, twist up into a knot in order to reduce its, entropy, or right, or to find its, minimum free energy state. Yes?
Yeah, yeah.
That's what it'll do. Okay. And so, by the way, just because it's now come up, those AI programs that are able to find the structure of proteins, uh, probably, though I haven't thought about it much, they can also do a good job on naturally occurring uh, RNA sequences. Now, for the same reasons, though I'm not sure of it in this case. Okay, so you got this necklace. When you throw it into salt water, it all coils up and acquires a three-dimensional shape. Now, it is true that it might get degraded by other molecules inside the cell, um, but sometimes it persists for a while inside the cell. I mean, uh, even uh, ribonucleases presumably take messenger RNA and degrade it. Is that right? I mean, I assume that's what happens to all this messenger stuff.
Well, so mRNAs, the cells have evolved this like mechanism where they put tails on the either like end of the RNA so that it
Yeah.
protects it from that. Um, but yeah, if it's in there long enough, it can still be degraded.
Yeah. Are you talking about like the start codons and stop codons and things?
No, so it's actually called a poly A tail, and then
Oh,
I think it's a G GT cap at the 5' end, the other end. Martina, correct me if I'm wrong.
Yeah, you're right.
Yeah,
okay.
Martina is my roommate, by the way. We're taking this class
Oh,
together.
okay.
Um, but
All right.
yeah, so those actually protect it from those RNases, and like basically the poly A tail is this long string of adenines, and it protects because the RNase will just like chew on this A until it's expressed, and that's like the mechanism for protection.
Well, but basically, it seems to me what you're saying is the cell can control both the RNases that are in there, in the cell, and the information it puts into the RNA strand in its sequence, and thereby the genes themselves have determined how long a particular sort of DNA will survive in the cell. So they largely control that. If they want a particular form to last a long time, then they have to design it to avoid whatever RNases they also design will act on it. So they don't just degrade, they degrade, I think, in terms of uh, a rate which is determined by the cell. Okay,
Yeah, I
so
think that's yeah.
yeah, all right, good. And so, um, what do the RNAs inside the cell do? We know that the messenger RNAs go find a ribosome and you know encode and cause a protein to be made, and then eventually they degrade by some mechanism that's in the cell. But there's a lot of RNAs in a cell. What do some of the others do?
So I mentioned a ribosome, so that's mostly protein, but it actually consists of ribosomal RNA. So RNA
Yes.
actually is part of the ribosome. There's some called silencing RNA, and there's a whole different like subset of those which basically act as like a silencing mechanism and will bind mRNA for degradation to like
Mhm.
silence some genes. And
Yes.
then there's very new one that's coming out. It's called long non-coding RNA. That's pretty mechanism is not exactly known as to what it does. I believe I'm missing. I mean, in bacteria, there's guide RNA for CRISPR, the gene editing system that guide this
Yes.
protein to certain locus. I might be
Mhm.
missing a couple types. But yeah, those are the main ones, I think.
With
Okay.
tRNA as well.
Oh yeah, tRNA. Yeah.
Oh, tRNA, yes. Okay, but let's consider the RNA inside of the ribosome. What's it doing there?
So it actually helps recruit the ribosome to the correct like portion on the mRNA. And so it's a very important part of like the catalytic process. It also helps bring together these two subunits of the ribosome, so that it actually like sandwiches the mRNA.
Yeah, I guess it does all this. Here's one thing that RNA does do. Uh, it can act enzymatically. That is, just a piece of DNA can acquire a shape where, for example, it will cut uh, um, pieces of DNA just by itself, not by recruiting other things. It can actually have catalytic sites on it that catalyze things, including, helping, I guess it's hydrolyze DNA, certain DNA sequences. Are you aware that it can do that or no?
Yeah, they're called ribozymes. Yeah.
Yeah, ribozymes. Yes, of course. Right. That's the word. So, okay. Catcher and Martino, you guys are aware of ribozymes. Yes.
Yeah.
Okay, you will answer for Martino.
Oh,
Okay.
I took my head. Yeah, I was saying.
Yeah. Okay, that's what I want to talk about. These RNA sequences curl up into a particular shape, and in that shape there can be a catalytic site that can, for example, cause it to take action or catalyze um, a reaction that, for example, other RNA strands, uh, it may cut them or do something to them. All right. With that as the basic, uh, I want to consider
Consider a hypothetical RNA like early Earth that consisted of an externally energized open system containing a G, U monomers in an aqueous solution, and which returns heat to its environment.
Okay, so my you know, thermodynamics and physical chemistry are not really very sharp, but um, if you had a pool full of random RNA strings and it was in an open system, there's energy and maybe will allow matter to also come in and go out. Okay, so, and there are a lot of RNA sequences and a lot of RNA monomers. Okay. And so let's assume that those monomers.
Further, let's assume that the monomers can form heteropolymers because when the local conditions are suitable, a monomer may attach to the end of a polymer to make it longer and reduce the number of monomers by 1.
So I'm saying some of these ribozymes might just acquire the catalytic ability to catch on to an arbitrary other ribozyme and chomp off one of its monomers. Okay. And um, and so then it frees up one monomer and it makes the monomer that got chomped on shorter by one monomer. Okay. Okay.
Heteropolymers because when the local conditions are suitable, a monomer may attach to the end of a polymer to make it longer and reduce the number of monomers by 1, and when local conditions are suitable, a monomer may detach from the end of a polymer to make it shorter and increase the number of monomers by 1. Let's further assume that among the polymers there arose a catalyst. This is known to occur when the polymers are RNA molecules. I don't mean that a polymer arose that could self-replicate, that is a very sophisticated form of catalysis.
Okay, so this monomer just by randomness arises, and it has this property.
Catalysis, but could do simple things of the sort that we know RNA can do. For example, what if some polymer, call it a P-polymer, emerged that could act catalytically on polymers that ended in AAA by chopping off the last A? Let's further assume that P-polymers have lots of A's but do not end in AAA. What would happen?
Okay, so anybody, but in particular, Catcher and Martino, with this setup, what should we expect to happen? So this polymer arises, it's in this RNA-like world.
Yeah, I understand. I think what would happen is like competition, quite simply, as in this P polymer would start hydrolyzing, as you want to say, or like attacking the other
Yeah,
polymers that end in AAA, but it wouldn't hydrolyze itself because it wouldn't be able to recognize itself as a potential target of hydrolysis,
right.
catalytic activity.
Yes, and so let's observe the um, P polymer through time. Every time it sees another polymer that ends with three A's, it chomps off the last A. Okay. So this should make the polymers that end in three A's unhappy because they're losing an A. That is to say, all polymers that end with three A's are being attacked and their copy numbers are being diminished. At the same time, lots of free A's are produced, and the P polymer happens to have lots of A's in it, but it happens not to end with AAA. Okay, some fixed thing that happens to that property. So, what would eventually happen if we look at this system over time? What about the copy number of P polymers? What would you expect to occur?
Increase.
Hmm? Yeah, I'm sorry.
It should increase.
Yes. Okay. It should. Okay. And on average, what's going to happen to polymers that end with three A's? What are their copy numbers going to do?
Decrease.
Decrease. Yeah. So let's take a break and we'll come back and explore this P polymer some more because, yeah, we'll see why. All right. So we'll take a break and okay, let's see. Stop sharing, I guess. All right, and I'll see you in 10 minutes, 3:03. Hello. All right. So we're talking about our catalytic world, and we're talking about our P polymer. And for you computer scientists, you know, you don't have to think about uh, the folding and everything. There's just a bunch of sequences or words over a four-letter alphabet. Some of them can eat the others in a certain sense, like the P polymer one. Uh, Whenever it's comes in contact with something that ends with three A's, it lops off an A, okay, etc. But uh, but if you can think about it as this RNA-like world, that can exist, and we know RNA can do things like what we just said. Okay, so we get
The
to
catalytic action of the P polymers would increase the number of A's in solution, which would then enhance the probability of forming new P polymers. It's true that the number of polymers that end in AAA would decline, but that is not the P polymers problem. The P polymers would have struggled for resources with the polymers that end in AAA and prevailed. Of course, if P polymers can exist, then presumably other catalytic polymers can as well, and perhaps a new polymer would emerge that increased its copy numbers at the expense of the P polymers. It seems that an evolutionary struggle has begun.
yes, okay,
Begun. So, simple catalysis has led to an evolutionary struggle. I believe that this is the same struggle in which self-replicating molecules would eventually become engages, cells would eventually become
engaged.
engaged, and all currently living things are now engaged. So, when did life begin? You might say that a thing is alive if and only if it can reproduce, or if and only if it has DNA inside a membrane. There are a huge number of possible criteria.
So, um, yeah, here's the question. A question. Let's talk about this P polymer. Does it reproduce? Okay, any answers that anybody would like to articulate so we can discuss this?
So, I mean, I seem to recall that before the break, in the introduction of the P polymer, you mentioned that it is self-reproducing.
No, no, no. It's not a self-replicating molecule in the sense we think of, one that can catalyze a construction of a copy of itself. That's a self-replicating molecule. It is catalytic, but it can't do that. I was saying self-replication is a really sophisticated form of catalysis. Okay. You know, you can catalyze from the material in your environment another copy of you yourself. That's really tough. These guys can't do anything like that, right? They can do just simpler things like bite off A's, right? But the question I have is, we have seen that the copy number of the P polymers is rising. Yes. So there's more P polymers if we look through time. Yeah. Now, uh, if we were watching cells and we look through time, we might say, oh, the number of cells with this gene set is increasing. Yes. And we would happen to know that the mechanism of that increase would be the division of cells and the stuff we learn in biology. But the end result would just be an increase in copy numbers for those cells. Now we see an increase in copy numbers of this P polymer. Why wouldn't that be replication?
Seems more like propagation to me.
It's what, I'm sorry. Yeah. Catcher, was that you?
Yeah, it seems propagation to me, like as if it's having like kids per se, but it's not actually something bringing the formation up
Wait, so catcher, I think
perhaps.
you sort of have hit a little glitch in the uh, Zoom. Seemed to have frozen up. We'll get back to you. Okay. So the question is the P polymer replicating? Anybody else willing to take the opposing point of view? That, let's say I take that it is. Okay.
Well, I mean,
Yes, Martino.
it's like you mentioned, it's not creating copies of itself. So each copy of the P polymer is not using itself as a template for the construction of a new P polymer. The
It
new
is not.
the new P polymers seem to be spontaneously arising from local, be it dehydration reactions, that polymerize small nucleotides into a longer chain.
Mhm.
But the the template is not it's not a replication because it's not using an existing P polymer as a template.
Okay. All right. So let me ask the question, um, which is who cares whether it's replicating? Because replication is one way to increase copy numbers. And it's a specific way, this template idea. But that's not what the struggle for survival is all about. It's much more about increasing copy numbers. So I would suggest that we may not really care if it's replicating in the sense, reproducing in the sense of the template model. Doesn't matter. That's just one way to go about it. The P polymer doesn't go about it that way, but it produces the same, more important end result of increasing copy number.
I understand. I think I see what you're saying, which is, and correct me if I'm wrong, but what you're suggesting is that by depolymerizing polymers of the same made up of the same monomer as itself that
Yeah.
aren't of the same primary sequence as the P polymer is kind of ensuring that any new forming polymer of nucleotides adopts its primary sequence because it's basically hydrolyzing anytime a new polymer is formed, it's hydrolyzing, um, in this case, a triple A tail, um,
Mhm.
into something that resembles more of what the P polymer itself looks like. So in this way, instead of directly replicating itself, the P polymer is kind of manipulating the formation of new polymers by preventing the formation of polymers that aren't mirrors of itself or similar to itself.
I don't, I think that's too strong for my taste. Okay. So that's it. It, in my view, the P polymer isn't um, trying to stop the formation of any other polymer. What it does is if a polymer has been formed and it has three A's on the end of it at this point in time, and it encounters the P polymer, one of those A's is going away. And that has two impacts, at least. One is that the P polymer, by assumption, has lots of A's in it. So if there's more A's floating around in that solution, it increases the chances that another P polymer will be formed than before there were so many A's. That's all it does. Okay. And it also, if we now look into the reaction chamber and count up things, we see that there's a noticeable lack of P polymers that have 3A's on them. And there's a little boost in all the molecules that happen to have a lot of A's in them, but don't have 3A's at the end. And so what we're talking about is basically the concentration of the P polymers and other things that need a lot of A's is sort of going up. And the concentration of things that happen to end with 3A's is going down. Yes. But the end result is that uh, there are becoming, through time, more and more polymers that don't have 3A's but need a lot of A's than if this was just a random collection of polymers, then we'd have in that case.
I see.
Yeah. So, okay, John, please.
Yeah, I was just wondering, this is not going to be very precise, but do you see a cause for the struggle to survive? Or do you see things happening sort of at random and it happened to follow a path where we observe the struggle to survive?
Yeah, here's the way I sort of see it, but I want conversation because how to see it properly is still somewhat elusive to me, though I have some hints. I don't think that there is any sort of global intention to have a struggle to survive. I think it's a consequence of the laws of physics. Our universe just has these things going on, okay? And that's what's going on. So that we know that in a system, an open system, and I don't think I have my thermodynamic chops good enough to do this right. We know that uh, it is okay. Let's see, how should I go about this? Okay, the main point is there's no intention to have a struggle or evolution. It's just what happens when you have a universe like ours. Now, the so it one could say that I think it's just a law, it's another law of physics. That is, if we observe the universe enough, we see this stuff a lot, we should recognize that this is what happens in our kind of universe. And I'd like to understand that better. So let's talk about this last sentence. So we'll do it again. Okay.
So, when did life begin? You might say that a thing is alive if and only if it can reproduce, or if and only if it has DNA inside a membrane. There are a huge number of possible criteria. One criterion that I prefer is due to geneticist Hermann J. Muller, cite, the gene is the basis of life, and physicist Edmund Schrödinger, cite, what is life, both Nobel laureates.
Okay. So Schrödinger is probably the more familiar of those two, he of the cat and of the Schrödinger equation of quantum mechanics. Okay, so he wrote this paper. It's about 30 or 40 pages called What is Life? And he tried to take a physicist's point of view of life. And by the way, Herman J. Muller did the same thing even earlier, but he got less attention. Okay. And he basically said, what I'll write here is my interpretation of what he's saying. His is a very interesting article because it really illustrates how confusing it was to a physicist like Schrödinger, how life could do what it did. That is just didn't seem to make sense to him, and he thought about it a lot. And in the 1950s, he produced this book, which here's my interpretation.
In my interpretation of their ideas, a thing is alive if it can maintain a concentration, roughly, copy number per unit volume, that exceeds what it would be if the system were closed and allowed to reach equilibrium.
Okay, so what am I saying there? That is, if you if you have a chemical reaction going on in some beaker, uh, it will eventually reach what is called equilibrium. It's also the place where the free energy drops to zero. Yes. I mean, if any of you are really good at thermodynamics, help me out here. Okay. All right. So now, if we look at the Earth, for example, oh, and this, by the way, is the reason for the prediction that the universe is going to end up in a, gray mist of some kind. Yes, because eventually the universe, being a closed system, must reach this equilibrium, the lowest free energy. Okay. So, but if you look on Earth, there's a heck of a lot of chemicals that seem very far away from reaching equilibrium. For example, sugar. Let's take glucose, right? At equilibrium of our planet, if it ever got there, how much glucose should we expect there to be in the resulting soup? Yeah. Nathan?
0.
Exactly. Okay. But it isn't zero. And physicists have been aware of this for a very long time. And it has to do with, I guess, free energy. Um, if you're looking at chemical reactions, you have these reversible reactions. These constituents will go to those and at a certain rate forward and a certain rate back. And at equilibrium, that will tell us exactly the concentration of each of the chemicals. Let's say it's something simple, but anyway, each of the chemicals, the concentration of them all. Are you familiar with that from some chemistry sort of thing? You know, you get at equilibrium, there's a certain concentration of every molecule. And that's determined by the forward rate at which those molecules are reacting and the backwards rate of those reactions, which undo what's been done. At equilibrium, that tells you the concentration of all the molecules in your system. But when it's not at equilibrium, if the system is open and there's a source of energy, it can actually result in you being very far at a particular moment from the equilibrium value. So the amount of glucose in the world right now is very far from the value it would be if we suddenly closed the world system. If we put the world in a box, closed it off to all interactions with the surrounding environment, then it will reach equilibrium. Now, if we close it off entirely, it will reach an equilibrium. If we close it off but allow heat to be transferred between the environment and itself, then it will reach a different equilibrium. Okay, so what Schrodinger is aware of is this notion of entropy, that systems tend to become more entropic, more disordered. And when they're closed systems, they will become less and less disordered until finally they reach a point where they cannot become any more disordered, and that's equilibrium for them. Okay, so so whenever you see a concentration of a molecule that's above what it should be at equilibrium, it's because energy is being acquired from the outside world or material, and it's being used to overcome entropy. Just like you make your bed every day, because if you don't, it's just going to become, or you clean your house every once in a while, because if you don't, it just becomes more and more entropic, right? Stuff falls apart. You have to put in energy to overcome entropy, but it's well known you can do that. But you need to be an open system where at least energy enters in order to do that. Okay, so as far as I can see, the P polymer seems to meet Schrodinger's definition of what it means to be alive. All right, so yes, so there's a lot more to say about this, and maybe we'll get to it. But here's my suspicion.
I suspect that an externally energized open chemical system at a modest temperature would have to be extremely simple to avoid this kind of phenomena and fail to have living components. Perhaps, this is the case with some non-chemical open systems as well. Consequently, I suspect that life is ubiquitous in the universe.
Where this is the definition of life, right? I mean, it doesn't mean intelligent life, but it can be as primitive as the P polymer. But you're going to find it every place. Hard to avoid. If you got a planet that's revolving around a sun, you've got an open system. Yeah. Okay. So what's your take on that? What if we have a planet? Well, just in general, any takes on all of this?
I have a question. So,
Yes, thanks, Martin.
when you're referring to open and closed systems, are you referring to kind of the traditional physical definition of transfer of energy, or are you talking about transfer of matter between inside and outside the system?
Yes, so that's the difference between what's called a closed and what's called an isolated system.
Precisely, yeah, but the Earth and any planet is an open system. I mean, besides
Yes,
the occasional asteroid that lands on Earth and introduces a bit of matter, we have energy coming from stars in a
yes.
form of electromagnetic radiation, and that makes us an open system, which is why. Without the sun, there would be no life because the sun provides the energy that, like you mentioned, we use to decrease entropy, whereas the second law of thermodynamics would state that it would always increase. So,
Mhm.
So when you say a planet is a closed system, I don't understand what you mean because
Oh, I never said it nor intended it, Martino. I say it's an open system. Uh, but of course, primarily, that is, it can receive the asteroid and it can receive energy from the sun. It can also send energy in the form of heat, usually back to the universe. And it can also, presumably send rockets out into space. And so it goes both ways. Uh, it's an open system, but we can imagine what would happen if we suddenly close the system. And closed means we'll still allow heat transfer to the environment or energy to be right. And um,
does not matter.
yeah, not matter.
Okay.
But actually, it's probably, is this what's called an adiabatic system where we only allow heat transfer?
I
Um,
think so. No, I think
wait,
it. Yeah.
adiabatic is it's the opposite. It's when heat is not allowed to transfer.
When heat is not allowed to
Yeah,
transfer.
so an adiabatic expansion, for example, is an expansion of gas, but the temperature of the gas stays the same.
Oh, okay. So what do you call in the systems where it's basically an isolated system, except you allow heat energy to transfer between the system and its environment? Is there a name for that one?
I could look it up.
I
I don't
should
know.
Yeah,
remember this. Oh no.
yeah. Please look it up. That seems like the system I want to talk about. So what that allows, of course, is that the temperature will eventually equilibrate from within the system's temperature and its surrounding temperature will eventually equilibrate. Okay, well, while you look it up, um, there's there's more to say about this. Um, Isabel, did you have something to say about it, or
Yeah, I had a question. Because
okay?
I just started reading the Schrodinger thing. What is life? Because of our last week's discussion, I
Yes.
was I was wondering, does he ever talk about like active equilibrium? So like when it's like an open system, but it's still able to maintain equilibrium. Because like I would say that's more akin to life than yeah, but it he
Yeah.
doesn't make a distinction between like closed system equilibrium and open system equilibrium.
No, he isn't that direct,
Mm.
or because he's trying to speak to a larger audience. Whether he could, in fact, be more direct in what he says, I'm not sure. I mean, I'm sure he could put it in other language for real physicists, but I don't know if he gets to the bottom of what is life. I mean, he's got the idea that life is something that has some free energy in it.
Yeah.
And when that free energy goes away, ain't no more life. I
Mhm.
mean, that's one way to say what he's talking about. Um, Martino, is there a name for my system?
Yeah, so you were asking about a system that has like physical or chemical reactions inside in which energy is is able to be transferred between the surroundings and the inside of the system. Well, that would be um but not matter, you said, right?
Yeah, but let's leave matter out.
Okay, so well, that would be a closed system, and then the processes inside of them would either be isochoric, isobaric, or isothermal. But they all three of those would involve the transfer of heat energy. I know it sounds counterintuitive that an isothermal process can have transfer of heat energy, but
No,
even
I think it would. Yeah.
but even though the temperature stays the same, in order for, say, a gas to expand, it does work on its surroundings, and so
Hmm.
energy is required for that to happen if you maintain the same temperature. Um, so, and then the only process that doesn't involve energy transfer would be an adiabatic process, which is what we were.
Okay. Okay. All right. So thank you for that, Martino. I don't think I'm going to understand exactly, the thermodynamic properties I'm talking about. Um, but but maybe you could, Martino, or others of you, and can, maybe actually explain this. That'd be a term project. Okay, but I want to say more about this. Um, let's say we're in an open system and the sun and the earth will do that. Now, there's all sorts of radiation that comes from the sun. Some of it's electromagnetic, right? Okay. Now, you get zapped with electromagnetic waves. I think X-rays are an example of an electromagnetic, aren't they? Okay. So if a cell gets zapped with an electromagnetic an X-ray, um, what usually occurs within the cell um, when that X-ray hits? Probably nothing, but what can happen?
Like damage?
Yes, damage, right? It can blow apart, DNA, it can blow apart proteins, presumably it just, rips through and tears things up. It looks to me like it increases entropy in the cell. Yes? But here's an important thing. If the cell happens to be a plant cell, then and the light happens to be in the visual spectrum, and I guess it's what should it be green or something? Then what happens when it falls on that cell?
ATP or right?
Yes, ATP, right? Because the cell has evolved this chloroplast system, which is extremely complex, and its job is to catch a certain electromagnetic frequency or a range. I don't know exactly how it works, and use those photons to put together ATP from probably a DP and a phosphate. Yes, Martina.
This is a technical correction. Probably,
Yes.
I mean, you're absolutely correct that photosynthesis ends up in the end result of ATP synthesis, but it's indirect in the sense that the chloroplast creates a high energy electron carrier called NADPH, which is then used to synthesize anabolic or produce anabolic reactions that make sugar. And then that sugar is actually broken down in cellular respiration the same way
Ah,
bodies do it. And then
okay.
those sugars are broken down to make ATP. So the plant cell kind of has two functions. It takes the light energy, it harnesses that energy in the form of a actually unstable molecule called NADPH. That
Yes.
molecule is unstable, so it has a high free energy. It wants to pass on those electrons to a different molecule. And so
So it can lower its free energy. Yes.
exactly. So then the plant cell synthesizes a sugar, which it basically transfers the energy, uses it into making a sugar, and then that sugar is broken down catabolically to make ATP.
Thank you.
I had a question. Is that
Yeah.
process like reversible do you remember by any chance?
Sorry, is what reversible? The photosynthesis or the cellular respiration?
Like the breaking up of the sugar?
Yeah, that's called gluconeogenesis. And it's when the opposite happens. So you take pyruvate, which is the end product of the breaking down of glucose, and then you basically make it thermodynamically favorable for the enzymes to act in reverse. And then you go from kind of the broken down, you invest energy in the form of ATP. So you actually break ATP up, and you basically reform the glucose from its broken down components.
Yes, so it's reversible. Yes. But of course, in the process of getting reversed, you're going to have to give off some heat, I think. But at any rate, my point is that in general, these electromagnetic waves are increasing entropy in the cell and disorder in the cell and leading towards death. But the things that don't die are the things that can capture some of this radiation and store it in a battery. Whether we call the battery ATP or something else, doesn't matter. They can store it in a battery. And that's the ability then it's that energy that can be used to reverse entropy inside the cell. Yes? And that may be the, you know, sine qua non of a living thing, that it's able to take the energy it receives and store it in some kind of battery. And once stored in a battery, it can be used within the object to reverse entropy. Yeah. So that leads us to an interesting view of life. It's not just that it's able to maintain less entropy than one would think it should have, whatever exactly that means. This has to do with what it would have at equilibrium, whatever that means. But it's not only able to reduce its entropy, it presumably has to have a particular mechanism to accomplish that. It's got to have a battery. So anything that's living, I suspect, needs to have a way to take the external energy, it's in an open system, and use it to make a battery. So how is it that batteries evolve? That is, uh, take plants. They're able to take this certain frequency of electromagnetic energy and actually not just have it rip through and increase entropy. They're able to store it in a battery and then use that battery power to decrease entropy in the cell. How does such a thing evolve? How does that happen? Why did it happen? I mean, it happened to plants a heck of a long time ago. But what were the plants doing before they had batteries? Another way to ask this is, are there any cells that have existed historically that didn't have batteries of some kind? Um, if you have a non-plant, oh, if you have a non-plant cell, then it acquires energy by eating things that have batteries. It essentially eats their batteries, right? But it raises the question of what were the batteries that were available to living things before plants came along? There had to be batteries, according to this sort of thesis that I'm putting forward. Okay, Isabel.
Did viruses come before plants?
Oh, yeah, that's a good question.
Uh,
Um,
Viruses are obligate parasites, which means that viruses can only exist if there is a cell that is capable of replicating the proteins that make up the code of the virus. So think of a virus as it's kind of a shell of proteins with inside some genetic information that can be transcribed by the machinery, like the ribosomes of the host cell. So the virus cannot replicate without a host cell's organelles, basically. Yeah.
But
So
yeah.
uh,
So oh, please, Martina, go
the
ahead.
virus cannot exist before a cell can exist.
well,
Um, yeah, gotcha. Sorry.
no, I was just gonna mention that it was about plants, and not necessarily there could have been cells before plants theoretically, and then the viruses could have existed. I mean,
Yeah. So
if we're talking about plant-specific viruses.
yeah.
Mhm.
But to answer Professor Adelman's question, in order to eat or have a battery or a source of energy, any cells
Yeah.
before presence, like single-celled organisms would have had to eat other cells through phagocytosis, in order to acquire their molecules into, within their own cell body.
I buy that, but what it raises the question for me is there has to be something that starts
Mhm.
with a battery. And we know plants, we understand their battery. What about the battery? What batteries preceded plants' batteries? Okay, Nathan?
So I could be getting this wrong, but I think the very first life was some type of bacteria that lived in like a sort of hot spring. So it was just getting like geothermal energy. And like if
Oh,
it literally just stays there, it might get the energy from Earth.
oh, I like that, Nathan. It's a good idea. So, these extremophiles, they're sometimes called, or they used to be, they live in these thermal vents, in the Marianas Trench and stuff like that. I presume we've all seen these things or seen things living down there. And one of the ways they do it is the Earth seems to be giving off these sulfur compounds that have energy stored already in their molecular bonds. So the Earth has put its energy, from the core or whatever the geological processes are, into these molecules. That energy has formed these molecules, and those molecules then come up, and these geothermo, dwellers, then consume those molecules. So those molecules are little batteries, right? That is, they have energy in them. And if you can take them in whole, then you can exploit that. Wow. Nathan, I think you brought us to a deeper understanding of this. So, so now we're saying that it's the geothermal processes that are producing little batteries in the form of little molecules that in their chemical bonds have stored some energy. And then organisms see all these batteries floating around, early ones, and start to use that energy to do their thing. And that's really makes sense to me. And so eventually you get all of these molecules, and they or these organisms, and eventually you get other forms of batteries. Yes. Oh, I really like it. Good work, Nathan. Yes, Catcher?
Yeah, I was gonna add on to Nathan, and we were talking about like RNA possibly being like the first like replicating uh molecule. Um, that's like part of a bigger like hypothesis called the RNA world hypothesis. And
Yes.
part of that
Mhm.
hypothesis is that that actually occurred, like the formation of the ribozyme and everything occurred in like those hydrothermal vents. Because that provided the energy to like if you join two monomers, two nucleotides, you need a little bit of energy to form those
You
bonds.
do.
So they think that it formed like where Nathan was talking about in those vents or another like freshwater warm pool.
Yeah. Now I'd like to explore that some more. I'm aware of the RNA world hypothesis, but now you're saying that when we have, like, one way to say what I'm going to ask is, so the P polymer you you're suggesting in order to go and bite off these A's, well, that may be an exothermal reaction, but at least the construction of new polymers, one or the other has got to be require some energy along the line. And so you're suggesting that the energy comes from some molecules that have it stored in their chemical bonds that that actually, if I want to make the P polymer argument, ultimately to do the things I've attributed to it, it's got to be exploiting energy that it gets from someplace, sometime.
Yeah.
So, yeah, Martino?
Um, yeah, exactly. Kind of bouncing off what Catcher said, this chemosynthesis or kind of coupling the breakdown of one molecule to power the formation of another molecule
Yes.
or a polymer, um, is it requires a source of initial energy to form that original molecule. And even before, and the hypothesis for even before this kind of RNA world hypothesis that suggests that the isothermal vents were the
Hmm.
source of the initial energy, there are scientists who also have done experiments where they take a pool of water and they have different basic compounds inside of there. So, for example, like ammonia, and then they strike lightning on into the water, and the energy from the lightning is enough to spontaneously synthesize larger macromolecules.
Yeah, this is the famous urea.
Yeah, the
Urea.
famous urea experiment. Exactly.
Yeah.
So, I guess the theory even before the RNA world hypothesis was able to happen is that initially the macromolecules that enabled those energetic bonds to be stored in the first place, those macromolecules were formed through the spontaneous kind of like lightning reactions that happen
Yes. And
when
and so in
yeah.
particular, I'm sorry, please go on, Martino. You hadn't finished.
No, so and I guess you right now the one question I had earlier is when in
Wait,
this
wait, wait. Then before you go on,
okay, yeah.
um, it, it, one of the things that you can form with lightning is the amino acids sometimes form. Now, do amino acids store energy?
Yes, because they have bonds, and
Yeah.
so large macromolecules have energy stored inside of them as bonds. Um, However, the breakdown of those molecules releases energy, but there is an energy barrier called the activation energy for the
Yeah.
hydrolysis. And so the activation energy is not broken in like normal standard uh,
Right.
conditions unless you introduce like some like a small impulse of energy. The molecule is not going to spontaneously break down, even though it is at a higher energy state.
Yes, yes. And that's what catalysts are purported to do, is to give you a second's way to get around that energy barrier of activation.
Yeah.
a cheaper way. And yes, and so these RNA molecules that are biting off this A, and let's say that requires energy. Okay, so this ribozyme comes along and wants to bite off an A, and let's say that does require energy input to break the bond. Then presumably that RNA ribozyme not only attaches to the other RNA strand, but it also probably attaches to a third thing, which has some energy in its bond. And then it presumably creates a three-dimensional structure because the ribozyme is there that encourages that other molecule to use its energy to break the bond.
Yeah, so this is a lot of proteins work this way, and in our cells, for example, they use ATP. So
Yes.
the ATP will be hydrolyzed, and then that will cause the protein's shape to physically change.
Yes, of course.
As the protein is kind of forced into a new shape, it moves the two substrates that it's reacting, or in this case, it would be one substrate that it's trying to break apart to,
Yes.
to kind of make the local environment within the enzyme or the protein or ribozyme in this case favorable to create the products of the reaction. So exactly like you mentioned, it could be coupling one reaction that's breaking down a
Yeah.
molecule to another reaction that's building up a molecule.
Yes.
Yeah.
Okay, so
I
do you
had a
want to?
question earlier about
Yes.
this scenario that you were writing about with the P polymer. So in the scenario with the P polymer, are the new P polymers, the new copies, are they being formed by kind of these spontaneous introductions of energy through, for example, the lightning that we were just talking about in the urea experiment? Or is the energy being introduced by other chemical bonds?
That's a really good question. Yeah, so how are these guys being produced? It could be that they're being produced because these monomers, it's energetically favorable for them to polymerize. That's one possible answer, right? I mean, it could be. And that where you need energy is just to cannibalize them. That is to hydrolyze, if that's the right process, off the other ones. So maybe these monomers, it's energetically favored for them to form polymers. So you don't need anything. If you have the monomers, they're going to form polymers, but to break down a monomer may require energy. But now you have these polymers that might act catalytically to break them down. Does that work?
Yeah, my only kind of doubtful um, question for that is that we know that obviously spontaneous thermodynamic processes tend to increase entropy.
Yeah.
And so it seems counterintuitive that the polymerization of, in this case, nucleotides, which decreases the entropy of the system, should be thermodynamically favorable. There would need to be very large enthalpy uh, decrease in order for it to be spontaneous with a negative free energy, because the equation is, I believe, delta G is delta H minus T delta S, which is entropy. So, and I don't know if the change in enthalpy for this polymerization is sufficiently negative to make the process spontaneous. Yeah.
It's it's a good point. It's a good point. Um, and I don't have an answer, but I do have this. Number one, I never saw the process of life or whatever the struggle we're talking about is uh, as clearly as you guys have allowed me to see it. And I hadn't realized that it seems to all be going back to just physics in the rawest sense. that is geological processes and, you know, electromagnetic radiation and just sources of energy, which are, you know, ubiquitous in our universe. And that's really great. And um, since it's time to end this discussion, I want to continue this discussion because I think you guys, you know, have so much better understanding of what's going on thermodynamically and chemically that maybe we can make more progress the next time. So I'll probably want to return to this, but we'll probably also spend a little time just going through the book as well. So anyway, thank you guys. That was super enlightening. Um, so I guess I'll see you guys next time. Goodbye.